Thanks very much for joining me today, Christelle. It's great to have you on for this latest episode of Charting the Best Course. In this episode, we're going to be talking about investing wisely in bioprocessing. First off, could you please introduce yourself for our audience and give us a little bit of a background as to your industry experience and your current role?
Sure. So thanks, Felicity, for inviting me to this, interview. So a bit about me. So I've been spending the last 25 years in the biologics CDMO industry, mostly in senior business development roles, but still always working very closely with bioprocessing teams, and also having the chance to support the launch of new innovative technologies, cell line development technologies, continuous manufacturing technologies during my roles at Catalent or Evotech. So today I'm wearing two hats. Through my own firm, which is called CD Biopartners, I'm basically advising CDMOs, biotechs, pharma investors on bioprocessing technologies, strategy, business model, partnerships is very wide. But it has the advantage to keep me very close to this whole industry and to know where the industry is going, what's actually hot there, and I can do that across a lot of players. And alongside, I'm also serving as a fractional chief business officer role at Prolific Machine, which is a US-based company using light to control cell behavior in biomanufacturing. So actually that combination is really nicely serving this topic that we're going to talk today. From the advisory side, I can see where the industry is going, where it's getting stuck, and actually Prolific is providing one of the solutions to those challenges. So to me, it's really happy to talk about those topics, wearing those two hats today.
Perfect. As mentioned earlier, we are going to be talking about investments into bioprocessing for this episode. So it's pretty common within the industry when companies are facing capacity constraints to just build a bigger facility. From your perspective, do you think they might be better served with investing in process intensification technologies instead of just building a bigger facility? Are there any key considerations that you could highlight for the executives who have to evaluate that decision?
Okay, wide question. So in my advisory work with CDMOs, with pharmas, I see that the reflex to just build bigger facilities has been fading. But still, the industry has not converged into one alternative. Currently, I'm seeing kind of three paths. I'm seeing like a small segment of pharma company going all in into fully intensified continuous manufacturing from upstream to downstream. I could mention like Merck KG in Europe, which has been speaking a lot about that and their efforts. I could mention the biosimilar player Sandoz, who acquired the Just Evotech site in Toulouse in France, which is full continuous, bioprocessing. But those examples, they are still quite rare. There's more a larger group of biopharmas who have been equipping their traditional sites to run semi-intensified processes. So those are processes where you add like a N-1 perfusion seed train to boost upstream productivity, but where you can still run a fairly classical purification train. So not a big change to the actual infrastructures. So you can still amortize your capex, but boost a bit the productivity. And there's a new segment new and third segment of company, if I may say, who are more and more interested into the foundation of the productivity itself. So basically looking into new cell line development technology, plus everything which goes around that, but to lift the titer early, to get a better process from start, and then, of course, they adapt all the upstream and downstream around that output, because if you produce more, out of your cell line, it may well that you have to adapt actually a continuous train to manage the output out of the cell line. But they are more focusing on the cell line first. So that would be a kind of a, let's say, three trends I'm seeing, but there's a lot of intersections and crossover between the three. Underneath all of that, one clear trend I'm seeing on new facilities is that, yes, clearly everyone is going towards lower capex. And to a degree to lower OPEX because they are starting to do more automation in-house, even if automation is not an easy thing today. A lot of people talk about it, but it's easier to talk rather than to do. The vast majority still of the industry, when designing new facilities, they are moving to smaller bioreactors, the 2,000 to 5,000 liter single use, instead of the old 20K liter stainless steel bioreactors. But back to the point that those smaller bioreactors, they can only meet large demand if you bring higher productivity cell lines in and high productivity process. So that's where you see this kind of evolution towards smaller bioreactor size, meaning smaller capex, smaller site overall, combined with this trend to go with higher productivity cell line, so that you've got the most out of those, let's say, trends. So if I may conclude there, for if an executive has to make a decision, it's always like fit for purpose. Not one solution is going to fit all supply, demand, every molecule. But currently, you can see that bigger stainless steel bioreactors can still bring cost efficiency for older, low productivity processes, which still exist, and for very large demand. But when you do that, you kind of lock the cost, and it's a pretty rigid setup, in terms of infrastructure. And actually, the highest leverage, the lowest disruption lever is really usually with the cell line, which is so playing with the cell line, which is the foundation of the process. And that's clearly the layer where we are focusing at Prolific to leverage light control protein expression, also called optogenetics, for those who do not know the technology platform, to boost titer and also create high-performing bioprocess, so adapted to also the complexity of the pipeline of biologics that we are starting seeing and that we're going to see more and more in the future.
Yeah, you mentioned about automation there. Obviously there's a lot of considerations to take into account, and it's not always a simple process. So what might a truly low-risk transition to a highly intensified process look like from an investment perspective? Can companies scale up yield without having to have an overhaul of their facility?
Okay, so here it's going to really depend on how far you want to go. So if you're trying to convert a traditional fed batch plant into a fully continuous perfusion-based one, you're essentially rebuilding the plant. So that's high capex, high risk, because there are still, some skills required to run that kind of facility. So here it's not low capex for sure. The low risk path is a bit of the opposite. It's really leaving the existing infrastructure in place and moving the productivity lever upstream of the hardware of the bioreactors, so into the cell line. So here, to take this low risk approach, low capex, you keep your bioreactors, you keep your process backbone, and you may add a bit of continuous downstream only if the output demands it, but actually you play with high performing cell line so that you get a better output from your existing capabilities. And again, talking about Prolific, that's exactly where the platform fits. Basically, the team has developed a light control cell line which can be manufactured into existing bioreactors, which we just need to equip with what we call illuminators. So they are LED, so lights, you just put lights into bioreactors, and which can already help to double or triple titers. It can help get a better pairing of multispecific molecules, control the glycans, and again, without having to rethink the infrastructure and go for a new footprint. And if we look at the pipeline evolution, it's going towards the same direction actually. The multi-tons of kilogram of drug substance that we saw some years ago for big blockbusters, the Humira and others, it's kind of behind us. The future is going to be made of ADCs, of multispecifics, all fitting a more precise indication, so smaller population of patients, which is going to mean smaller volume, but with actually a more complex format to develop and manufacture. So you're going to have really to rethink the capex, because you're going to have to adapt to more molecules, more complex one, smaller volume, and with a bit of more risk embedded of those pipelines. So back to actually the point, to setting up a flexible kind of smaller scale facility. Currently we're seeing a lot of facility with 2 to 5 kiloliter single-use bioreactors, and again leveraging high-performing cell lines so that you can meet both the clinical and the commercial demand with the same setup.
Yeah, as you mentioned there about the novel pipelines, we are moving away from those blockbusters to those unique niche, smaller yield products that have high value. So when introducing novel bioprocessing platforms, how might that affect the risk-adjusted net present value of a pipeline molecule? Where do we see the biggest gains? Is it in accelerating timeline to clinical trials, or in drastically lowering long-term cost of goods?
Okay, so talking just first a bit about the risk of new technology, because if I start there, I would say that the risk to me is less into the adoption of new technologies rather than with staying with like kind of old tools that actually are not going to work for the next generation of biomolecules, both from a developability and COGs perspective. So let's say maybe that's the first point. So if here I talk, for instance, about the prolific technology, which is new and which basically has many benefits. Again, our team has been doing extensive modeling and development to de-risk the scale-up of optogenetic cell lines. So basically to make sure that what is a great new technology, early scale, solo scale, earlier on, can really translate into commercial path. But it's true for any new technology. The zero risk approach does not exist. But again, I truly consider that the downside is going to be minimal and far smaller than the upside. Because if you take no risk, you're going to be stuck with old technologies, and you're not going to adapt to what is ahead of us. So in terms of biggest gains, I actually see two directions. So basically, you mentioned timeline. But also probability of success, because a lot of the existing complex molecules, we're seeing already a lot of multispecifics with pairing problem. There are some cytokines, toxic molecule, basically sitting in pipelines, killing the cells. So great molecules, but we can't basically find process that can manufacture them. We see a lot of fusion protein, which are actually stones to express, very, very difficult to get a proper yield. And a number of them, not even the new one, but existing one, they're already stuck in clinical development just because their half a gram per liter titer is not sustainable, or the process is too difficult to scale to move into a phase 3 and envision a commercial path. So actually a great CMC package with a higher titer, with a reproducible process, better chain pairing for multispecific, better control of glycosylation for biosimilars. That's also what's going to make a challenging program or a stuck program being developable, and make sure that investors, whoever they are, they can improve their return investment. Do not waste like hundreds of millions in early clinical programs, which will never see the market, and actually ensure that patients are going to see more and better treatments. So accelerating timeline for sure, because when you come with new technology, you actually unlock the developability, and you can spend months, years on developability. So it's both timeline to clinic and time to market, and basically improving the success rate. Secondly, the COGs. So, and here this is becoming existential. It was not that important in the past, but it's more and more. And again, I was saying, I'm seeing a lot of molecules that basically could scrape through phase 1 and poor yield, but now they are stuck in the middle of a clinical development because no one wants to invest in a molecule that promising it is, with huge market indication behind, but which doesn't have a sustainable CMC process, a sustainable yield, a sustainable COGS. This is not possible anymore in the current regulatory and reimbursement environment, as simple as that. So the COGS can kill the economics, and that what ultimately it's not yet palpable, but it will become more and more what is going to block the funding, the out licensing, and clearly the path to market. So on all the points, timeline yes, COGS yes, process reliability. We hear a lot also of product quality, process robustness. This is key for pharma to ensure the supply demand doesn't have hiccup at commercial stage. And again, this is exactly what we are trying to do at Prolific. Develop those high titer cell line with controllable process that can actually scale and that can address the manufacturing efficiency, the COGS efficiency from day one, and not look at that basically 5 years down the path when basically that can become a bottleneck to the commercial path.
Historically speaking, dynamic control of cell lines has relied on expensive chemical inducers, which can skew CMC data packages and downstream purification costs, downstream being a bit of a bottleneck as it is at the moment. We're seeing those high titers coming from upstream, downstream struggling to keep up. So from a CapEx and OpEx perspective, how does shifting the control lever from chemical molecules to physical inputs such as light or automation change the cost structure of a therapeutic program?
Okay, so maybe just a minute on your comment about the downstream processing, which can be a bottleneck. So clearly, this is very true. And that's why at the minute, more and more companies, we are looking at putting a bit of continuous processing in the downstream so that they can just manage the output. But this being said, you have to manage when you have big output. Reality is that today, for some complex proteins, you're not going to generate 20 grams per liter. You're going to still generate 5 grams per liter, like you were doing for standard mAb 20 years ago. But with the difference that those 5 grams per liter, they're going to be okay because it's going to be a cytokine fusion or a super tricky fusion where 20 years ago, you would have been at half a gram per liter. So that's where the continuous processing, again, will be needed for the large outputs, but a lot of those new processing, they're just gonna basically transform very difficult to express candidates into sustainable molecules with decent titer, decent enough to go to commercial, but without facing the 20 or 30 grams per liter, because that's also the reality. So back to your question on impact of chemical inducer cost, and so here I'm pushing back a bit on the premise. I really don't think the big COGS lever is to get rid of the chemical inducers in the final bill of materials. The inducer itself usually is not the biggest line. So where light, so at Prolific, the optogenetic control with light, it generally helps on the CMC side because you're going to get no inducer, and it's going to help to clear the downstream, and it's going to help also to better characterize your data package. But to me, that's even secondary. The real lever, it's again, it's really productivity and control. You take a difficult to express protein from 1 gram to 4 grams per liter, the example I was just giving. If you have like a decent commercial demand, you can save $20 to $50 million a year on the commercial COGs, just on the drug substance manufacturing cost, as simple as that. So basically the chemical inducer in that, it's not going to be the big play. And beyond the cost, I touched on it earlier, it's also the control. So actually controlling what's happening in a bioreactor can also help unlock some scale-up, which can be tricky for certain molecules, certain process. In tech transfer also, you can see some process change. You change the operators, you change the facility, the bioreactors, and something change. So actually having the ability to control what's happening live in the cells so that you can still hit your CQAs, your specifications, that's worth gold. And again, the industry is spending millions of dollars on failed batch. So this ability to control what's happening at your site at commercial scale, or when you get towards commercial scale, I think that's also very, very high value. So process consistency, product quality are to me as much important to the pharma industry than the timeline acceleration and the COGS aspect. You've got to combine them all.
And another aspect to consider is obviously that of personnel. As we're seeing, modern bioprocessing is incorporating more technological advances, so synthetic biology, physics, hardware engineering, and machine learning. How should companies invest in training and building their teams in order to handle these new environments?
Let's say that for any company building a new technology, training, skill building internally is not optional. To me, this is the condition for innovation to happen at all. Now, on the how you invest, so I think here modern bioprocessing sits at the intersection of synthetic biology, physics, like you said, hardware engineering, machine learning, and the mistake would certainly be to hire those as silos, so side by side. The value to me is created at the interfaces. So when you invest into those skilled people and make them work across boundaries, so basically having a biologist who can talk to the ML engineer, an engineer who understands biology, that's where you get basically the fertilizer to get the innovation happen. And this is how Prolific has been built. There's all, those skilled people working in their lab, but gathering together to make the magic happen and combine the different, let's say, layers of genetic biology, illumination hardware, AI-driven process control into one platform. Because they are working as a team. So, and I would add that, generally speaking, and again, this is true for Prolific or for any, innovative company, no one should never rest on what has been achieved. So you need to keep bringing new skills, new perspectives, and also cross-fertilize outward too. So basically speak with your partners, your customers, keep listening about their challenges, their ambitions, so that you can constantly, feed in the platform and generate the next, level of innovation, because, it's all moving. And we see that in our environment that, every 6 months, there's like new formats, there's something new. And this is also what keeps, training and keeping people, connected and wired to manage the innovation.
And looking ahead, what might differentiate the companies that invested wisely in bioprocessing today from those that played it safe? What does the ultimate smart investment look like in hindsight?
So here, and this is my really personal belief, I think the differentiator is going to be the ability to handle the new complex formats So unlock their development, accelerate their time to clinic and market, but combining that with a COGS reduction objective earlier on. Because if you really look at what's happening today, you see that to move fast, most clinical team at biotechs, they go with traditional toolkits, conventional cell line development, fed batch, da. The CMC consultants, they are very scared about using new technology because the goal is, okay, time to clinic, get your funding, demonstrate, the drug, the molecule is safe, and then we'll see what next. But what's next is that when those molecules, they are successful in phase 1, phase 2, they arrive at a stage where they've got to prepare for phase 3 commercial, pharma starting looking at them, and that's where you see that you've built a liability, so you arrive at later stage with a high value asset, but a poor CMC package, and everything fall apart. So that's where really I think the companies will perform, will win tomorrow, are going to be those, whether they are biotech or biopharmas or CDMOs, because they are all part of this ecosystem, that won't treat speed and commercial cost as a trade-off. That they basically combine all those aspects to pick up the best line, the best upstream, downstream approaches to unlock the complex candidate developability, accelerate, but develop something which is sustainable and which is high value, not only the molecule and its efficiency, but also high value from a CMC perspective. And everyone keeps saying that, all the CMC consultants keep saying that CMC is the key. And with the recent basically access to the FDA data, rejections data, you see that CMC was core to many failure to bring a product to the market, but still it has not fully translated in the decision made earlier on preclinical candidate. So again, that's a bit of a bet we are making at Prolific, putting all those elements within one platform, the control, the speed, the high titer to reach low COGs, so that we can enable our pharma partners, those who will have made the decision to combine all those aspects earlier on, to help them bring their pipeline to the next clinical stage and widen access to patients. Because at the end of the day, we behind the process, behind any product, there's patients. So that's what we need to keep in mind. So I think this is how I would conclude.
Perfect. Thank you, Christelle. It's been an absolute pleasure talking to you today.
Thank you very much, Felicity. My pleasure.